Greetings, we are Group 6 and our topic for today's presentation is Application of Bioinformatics in Lung Cancer Research. And here is our table of content. Now I'll pass on to Jingyi for the introduction of lung cancer.
A brief introduction on lung cancer will be shared before we proceed on the examples of bioinformatic tools used on lung cancer research. Lung cancer is the most common cancer in Malaysia and is the most common cause of cancer-related death in Malaysia also. Other than that, clinical trials on lung cancer treatment are limited in Malaysia. This, combined with the high cost of molecular testing, systemic therapy, and radiotherapy, limits the available treatment options for many Malaysian patients. Lung cancer can then be further categorized into two types, which is small cell lung cancer and the non-small cell lung cancer. Non-small cell lung cancer is more common than small cell lung cancer, and it is generally not that aggressive. Then under small cell lung cancer, we have two types, which is small cell carcinoma and the combined small cell carcinoma. Small cell carcinoma is the most common type of SCLC. The cells of a small cell carcinoma tumor look flat under a microscope. Then for the combined small cell carcinoma, it is a tumor that's made up of some small cell carcinoma and some non-small cell lung cancer cells. On the other hand, there are three types of cancers under NSCLC, which is adenocarcinomas, squamous cell carcinomas, and the large cell carcinomas. Treatment of lung cancer depends on three factors, which is the type of cancer, the stage of cancer, and the functional status and lung function of the patient. Treatment that could be done are the removal of tissue with cancer cells, chemotherapy which uses special medicines that shrink or kill cancer cells, radiotherapy that is using high energy rays similar to X-rays to kill cancer cells, or a combination of both therapy. Then there are also targeted therapy which treat cancer by interfering with specific proteins that help tumors grow and spread throughout the body. And lastly, the palliative treatment for advanced cancer.
Now let's talk about one of the database that was used in the research. GeoDatabase, which refers to Gene Expression Omnibus, is a database that archives gene expression and functional genomic datasets. It is created in 2000 as a worldwide resource for gene expression studies. Geo supports community-derived reporting standards that specify provision of several critical study elements including raw data, process data, and descriptive metadata. The database not only provides access to data for tens of thousands of studies, but also offer various web-based tools and strategies that enable users to locate data relevant to their specific interests, as well as to visualize and analyze the data. NCBI has divided its objective into three very specific terms, but it's created to make the data it contains accessible to the research community, and most studies are already analyzed by bioinformaticians and statisticians. There are three submitted records, which is platform, sample, and series. So these three records are organized into high-level categories for better analysis. The database stores all original submitted supplied platform, sample, and series record, as well as curated gene expression dataset records. Profiles, on the other hand, the database stores gene expression profiles derived from curated dataset. So let's talk about the interface of the GeoDataset result page. It is to note that the image on the thumbnail cluster image can be clicked to redirect to the dataset record. They contain several data analysis tools such as find genes, compared to sets of samples, cluster heat maps, experiment design, and value distribution. Moving on to profile result page, here is some of the overview of the interface. That's all from me, and then I will pass on the application to Amir.
So how is GEO used in lung cancer research? There's a 2021 study by Usman and Swaminathan where they identified the biomarker hub genes to determine the biological pathway and PPI network as the survival rate of patients are improved by this identification with bioinformatics tools. In order to access GSC 115456, GSC 2827, and GSC 84797, the geodatabase at NCBI was used to gather data for expression profiling. The raw gene expression data were processed using the GEO2R tool, which was integrated with GeoQuery, Bioconductor R, and the lemma package for analysis of the differential gene expression using tables ordered by gene and graphic representation of geoprofiles. The GSC 84797 dataset contained 28 patients with primary lung cancer, while GSC 2827 and GSC 115456 datasets contain 26 and 30 respectively patients with non-small cell lung carcinoma. There is also a 2021 article by Wang and others where they identify potential key genes, proteins, and associated interaction networks for the development of lung cancer in non-smoking women through a bioinformatics approach. They carry out the research using the GSC 1980 dataset, which contains 60 lung cancer cancer and related paracancer tissue samples from non-smoking women. The limit package analysis in our software was used to find the differentially expressed genes with the screening criterion of p-value smaller than 0.01 and log-to-fold change Fc larger than 2. The microarray was downloaded from the GEO database, and a total of 169 DEGs were chosen by assessing the gene expression microarrays using the desired criteria, including 39 upregulated genes and 130 downregulated genes.
So let's move on to David. David is a database for annotation, visualization, and integrated discovery. David is a bioinformatics tool that offers functional interpretation of lengthy genes lists acquired from genomic investigations such as microarray and proteomic studies. These are tools that is used by David to analyze gene lists. As well known, David offers a comprehensive knowledge base compiled from the most widely used bioinformatics sources. There are 4 complete tools that have been created to maximize the knowledge base, including functional annotation tools, gene functional classification tool, gene ID conversion tool, and gene name batch viewer. Firstly, based on the association between genes and annotation in the user's list, users can do functional annotation mapping, functional annotation enrichment analysis, and functional annotation clustering using the functional annotation tools. Secondly, the gene functional classification tool creates a gene-to-gene similarity matrix for the gene in the user's list based on the common functional annotation from several categories. Thirdly, gene ID conversion tools help you transform a list of gene IDs or accession to other identifier types. With these tools, users can also identify and determine the ambiguous or contaminated accessions in a user's list. And lastly, to observe the genes that are present in a user's experiment quickly, the gene name batch viewer may provide all of the genes names for a specific gene list in a short amount of time. Here is the web-based interface of David Knowledge Base. First, the Gene List Manager panel on the left side of the screen allows users to submit several kinds of gene identification. The associated annotation categories can then be selected and queried on the right side of the screen.
Thank you, Shafia. Now I will talk about the application of David in the study of lung cancer. We will discuss this from 2 research papers. First one is the meta-analysis and bioinformatic exploration of the diagnosis value of a molecular mechanism of MIR193A5P in lung cancer. The purpose of this study is to explore the clinical diagnostic significance of MIR193A5P. This highly conserved endogenous non-coding small RNA has been shown to be associated with multiple cancer. In this study, David was used in gene ontology and KEGG analysis. Here is the result of the analysis. These figures show the significant GO and KEGG enriched term of the overlapping gene. In other research paper that Amira mentioned before, David was also used to analyze gene ontology function and KEGG pathway enrichment. GO analyzed the function of large-scale genomic or transcriptional data, while the KEGG pathway analyzes data about molecular or gene integration network processes. Figure 1 shows the result of KEGG pathway analysis of lung cancer-related differentially expressed gene using the David online tool. Figure 2 is the result of the analysis of biological processes, molecular function, and cellular component using David online tool. Thank you. Now I will pass to Vincent.
Hello everyone, now I'll be explaining about the definition, the type of data shown, the aim, and the sources used for string database. Firstly, what is string? String is a proteomic database focusing on the network and interactions of proteins and a wide array of species. Strings allow the searching of one or multiple proteins at a time with the ability to additionally limit the search to the desired species. Now, what did the results of strings show? String results show visual representation of the interactions that the search protein is involved in, along with expression, location, transcription, and experimental data on the protein and its associated proteins. How about the aim of string? The string database aims to integrate all known and predicted associations between proteins, including both physical interactions as well as functional associations. To achieve this, string collects and scores evidence from several sources, such as automated text mining of the scientific literature, databases of interactions, experiments, and annotated complexes or pathways, computational interaction prediction from code expression and from conserved genomic context, and systematic transfer of interaction evidence from one organism to another.
Thank you, Vincent, for the wonderful insight. Now, I'm going to talk about how string is used in diagnostics of lung cancer based on a research paper by Piao in 2018. The research is intended to find effective biomarkers for early diagnosis of lung cancer because prognosis of non-small lung cancer still remains poor due to lack of effective diagnostic methods and high metastasis rate of the cancerous cells. This is done by constructing a protein-protein interaction network and survival analysis to identify the main genes involved in non-small cell lung cancer. The picture below shows the results of the PPI network, which was constructed using string. PPI network represent molecular complexes. There are 124 nodes and 314 edges in the PPI network. Nodes represent proteins and edges represent interactions. Next up, based on the results from string, 7 genes had high degrees in the PPI network. These genes played important roles in the progression of tumors. Past studies on these genes have revealed their functions or characteristics, such as ANGPT1 promotes growth of ovarian cancers and shortens the survival of experimental mice. CDH5 was overexpressed in lung cancer due to EGFR mutation. PTTG1 promoted migration and invasion in breast cancers. Finally, string analysis provide a comprehensive perspective to understand the mechanism of non-small cell lung cancer development by identifying the most important genes which carves a way to find possible early diagnostics or treatment for non-small cell lung cancer.
In conclusion, lung cancer is lethal since it is able to affect various organs in the human body in the form of carcinoma or adenocarcinoma. However, it can be treated with various radiation and drug therapies. The 3 databases that contributed to providing important information regarding lung cancer were GEO, David, and String. GEO was able to identify the cellular components, biological processes, molecular functions of gene expression, potential key genes, proteins, associated interaction network, and PPI network determination. David provides a comprehensive set of functional annotation tools for investigators to understand the biological meaning behind large lists of genes related to lung cancer through annotation, visualization, and integrated discovery. String was able to display visual representation of networks and interactions of proteins, particularly to analyze the association between protein-protein interaction and cancer. Lastly, these are the references, and thank you.